Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start!
In the previous video, we did about linkers and adapters. So we learned how we can use linkers and adapters to modify the DNA so that it can be easily inserted into the vector. In today's video, we are going to learn how to generate fragments for cloning. So there are different methods for generation of fragments for cloning, and we already did one of the most famous method, that is restriction endonuclease digestion. So anyone who hasn't watched the previous videos, just check it out. You have a proper explanation about what is restriction endonuclease, what are its types. So, just a brief explanation. Restriction endonucleases are enzymes that cleave the DNA at a specific sequence. Okay, target sequence. So they are endonucleases. So what are nucleases? Nucleases, they break the DNA, and endo means which breaks it in the interior. Okay, so I have brief explanation, a proper explanation about nucleases, endonucleases and everything. Do check out my previous videos for more proper explanation. So endonucleases, they cleave the DNA. So when this is, imagine this is a DNA which we are having, 5' to 3' and 5' to 3'. So I don't want this whole DNA to get inserted into the vector, and I can't even do it. That is when I use restriction endonucleases. So what does this restriction endonuclease do? They cut the DNA at a specific site. So I want this fragment, this specific fragment I need. So here this fragment has some sequences, okay, which are known as the restriction sites. And that is where the restriction enzyme site, they directly come here and they cut the DNA fragment, and hence we get the insert that is needed. So this is how the restriction endonucleases work. So this is one method in which we can generate the fragment. And the method is without using any enzymes, that is mechanical shearing. Okay, so this includes different methods. One, I can name it as sonication. So sonication is a process in which we are applying sound. So here you have application of sound, okay, which breaks the DNA into smaller fragments. The second is needle shear. So here we use a small needle gauge. So what does it do? It breaks the DNA into less than 20 kilobase pairs fragments. Okay, so here we are getting DNA fragments of less than 20 kilobase pair fragments. Now you have another method, that is nebulization. So what does this do? Here a fine mist is created. Okay, so how do you create a fine mist? We force, the DNA solution is forced to move through a small hole. Okay, in the nebulizer. So when it moves through the small hole into the nebulizer, the DNA fragments are being created here. Okay, and this is how you generate fragment. Next, you have point sink shearing. So in this point sink shearing, you are using hydrodynamic pressure. So hydro, as you all know, it means water. Okay, so here we are using the hydrodynamic pressure to create small fragments of DNA. Next is we are passing the DNA through a French pressure cell. So what is this French pressure cell? So here you are creating a high pressure that makes the DNA to shear or break into a smaller fragments. Now, remember, whenever you are using restriction endonucleases, you are getting a defined fragment. So here while using mechanical shearing, you don't get a defined fragment, you just get a lot of fragments of DNA. Okay, the next one which we are going to do is cDNA duplex from RNA transcripts. We are creating a complementary DNA. So what is this complementary DNA? Complementary DNA is the DNA which is obtained from the messenger RNA using a reverse transcriptase enzyme. This has also been explained very nicely in the previous videos. Just check it out. Here I'm going to give you a brief description about it. So what happens? Now I have an mRNA. Okay, imagine this is an mRNA. Now this mRNA encodes for a very important protein, which I want to be inserted into the vector. But it's not possible because this is an mRNA and my vector is a DNA sequence. So I need to convert this mRNA into the DNA, for which I use a process called reverse transcriptase. Okay, so using this process of reverse transcriptase, I can create a cDNA, which can be inserted into the vector after restriction digestion. Okay, so here I am creating this. This is the fragment I need. So I am creating it using this reverse transcriptase. Let me quickly explain. I have a mRNA with A tail. So mRNA, the most significance is it has an A tail. This mRNA, I'm going to add primer to this. So when I'm adding the primer, this is what happens here at the 5' end, the T tail, because it's a poly A tail, so you would be having a primer having T tail. So this is how your primer get attached, and then you add DNTPs and reverse transcriptase enzyme, from where you get a hybrid. What is this hybrid? Called as, this hybrid is called as DNA RNA hybrid or RNA DNA hybrid. Now we are going to remove the RNA from this, and then whatever is left, that is your cDNA, what is called as complementary DNA. Now we can also create DNA fragments using chemical synthesis. Okay, so how are we doing chemical synthesis? We are using a DNA synthesizer. This DNA synthesizer, it creates DNA by sequentially adding one nucleotide after the other. So how does this work? Let's have a look at it. So this is the process by which we are going to add the DNA. So step by step, let's learn about it. Okay. So artificial synthesis is mostly done in 3' to 5' direction. Okay. The first step is we will attach the nucleotide to a glass bead, as you can see here. Okay. This is a porous glass bead which is made up of controlled pore glass. Now, the nucleotide is not attached directly, but it is linked to the bead using a spacer molecule. Okay, so we have a spacer molecule here. Okay, now this spacer molecule that bonds to the 3' OH of the nucleotide. Okay, so as you can see here, this is the first nucleotide. Glass bead has been attached to it, and then it has been attached using a spacer molecule. Till here, I think most of the things are clear. Okay. Now, what happens? There are many beads that are held in the column so that the reagents can be washed and removed easily. That's the main reason we add the glass bead. Now, each nucleotide is added as its phosphoramide. Okay. Now, what does this phosphoramide do? It consists of a blocking group that protects a 3' phosphate group. So as you can see, we have a 3' phosphate group here. Now it protects the 3' phosphate group. Now, then you are going to add diisopropyl amine group, which is attached to the methylated phosphoride group. Okay. Now, before adding another nucleotide, here we have second nucleotide. Before adding the second nucleotide, what you have to do is you need to remove this 3' phosphoride, which is done by tetrazole, as you can see here. It's been removed. Okay, now then the next nucleotide is added to this. Okay, and it reacts with the phosphide to form a dinucleotide. This is the dinucleotide. Okay, now See, whenever you are in the second nucleotide, this second nucleotide also goes to the same process as the first one does. Okay, now the chain must be capped off so that it could prevent generation of incorrect sequence by later reactions. So the 5' OH is acylated with acetic anhydride and dimethyl aminopyridine. Now this terminates the chain. Okay, and no other nucleoside phosphonamides can be added. So this is how the direct chemical synthesis occurs. Now the next method is called polymerase chain reaction. So polymerase chain reaction is mostly used in most of the reactions, whichever you see, wherever lab you go, you will be getting that. So what does it do? It generates DNA for cloning, or I can say it amplifies the DNA. Okay, and then that amplified product can be just cut by the restriction enzymes and can be inserted into the cloning vector. Okay, so how does this PCR work? So PCR has three main steps, as most of you might be knowing. It is denaturation, annealing, denaturation, initial denaturation and final denaturation, and then annealing, in which the DNA binds to the primer, and then extinction, polymerization and extinction. Okay, I can write here polymerization also, polymerization and extinction. So this needs a particular temperature. The temperature is very important in polymerase chain reaction. Okay, and according to the product we need, we have different types of PCRs like touchdown PCR and asymmetric PCR and many more. These are different types of PCRs which are being used according to the function or according to the product we need. This was all about generation of fragments for cloning.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to BioDrunk. Let's meet in the next video. Bye-bye.